Good evening, guys, and today I will discuss about the glomerular structure and function, and this is the physiology. And we will also discuss the glomerular filtration rate and the basement membrane and the factors in the filtration. Okay, so first of all is the structure of the glomerulus. The glomerulus consists of in total 2.4 million nephrons, about 1.2 in each kidney, and each of the nephron functions as the filtration unit. Okay, after that, you know that glomerulus is formed of the capillaries, and capillaries are basically the afferent. They come into the glomerulus and they're drained by the efferent arterioles. So in between the area where the glomerulus is formed, it is the between the high pressure system. For example, when artery is draining into the vein, when artery is draining into the vein, it is a low pressure system. Most of the body anastomosing structures are arteriovenous anastomosing, but the glomerulus is an exception in which the glomerulus is drained by an arteriole, and it also the blood enters via the arteriole. So it is a high pressure system. And we need the pressure because we are requiring filtration here. So the glomerular tuft of capillaries is there. Also the glomerulus is surrounded by specialized cells known as podocytes, and they contain food processes. As you can see, this is a one cell of podocyte. It contains food processes, as you can see here, and there are two layers of it. First is the visceral layer and the second is the parietal layer. Okay, after that, the capsule is formed between the visceral and the parietal epithelial cells of the Bowman's space. It is known as the Bowman's capsule. This glomerular tuft of capillaries is surrounded by a connective tissue known as the mesenterium. It is a connective tissue and it contains mesangial cells. We will discuss the functions and properties of mesangial cells in the end of this lecture. Okay, after that, the basement membrane. Basement membrane, the glomerular basement membrane, is present between the endothelial cells of the capillaries and the epithelial cells of the Bowman's capsule. As you can see here, it is not mentioned in this diagram, but it is present between the endothelial cells of the capillaries. Okay, here, here is the location of the basement membrane between the endothelial cells of the capillaries and the epithelial cells of the Bowman's capsule. After that, the blood flows into the glomerulus via afferent arteriole and drained by the efferent arteriole. It creates a high pressure system which is specialized for the filtration. The main thing here is the fenestrations or perforations, which is that the endothelial cells of the glomerular capillaries have thousands of fenestrations, and these fenestrations size approximately 70 to 100 nanometers, and everything smaller than or is equal to 70 to 100 nanometer or in between can easily be filtered through the fenestrations which are present here, okay, in the endothelial cells of the glomerular capillaries. Okay, this is a structures you can appreciate here a lot of things. First of all, the arterioles coming and draining. After that, the mesangial matrix, the parietal layer of the Bowman's capsule, the visceral layer. This is the proximal convoluted tubule. This is the peritubular capillaries, and which is around the PCT and the distal convoluted tubules. And after that, this system is draining into the veins, ultimately into the renal vein. Okay. And the second topic is the glomerular basement membrane. First of all, the structure of the glomerular basement membrane is of high importance. Under the electron microscope, the BM basement membrane consists of the dense area. in the center, as you can appreciate here. I'll use the red color so it became more prominent. In the middle aspect, the glomerular basement membrane has a dense area which is known as the lamina densa in Latin, and around it there are less dense areas which are visible under electron microscope. These are lamina rara, and but this is the lamina rara interna towards the endothelium of capillaries, and towards the podocytes or epithelium it is the lamina rara externa towards the Bowman's capsule. Okay, what comprises the glomerular basement membrane? Of course, the collagen. Collagen, what type of collagen? There are four types of collagen present in the body. The collagen type 4, the triple helix of collagen type 4, peptide chains, and there are million of them which constitute the glomerular basement membrane. Let's discuss the collagen. First type is present in the bone. How to remember that is 1. So 1 means bone, bone 1. Okay, type 2 is present in the cartilage. Type 3 is present in many places in the body, and type 4 is mainly the constituent of the basement membrane. Either it can be the glomerular basement membrane, it can be the alveolar basement membrane in the lungs. Okay, and what a comprising of? It is comprising of triple helix of collagen type 4 alpha peptide chains, and these peptide chains, basically proteins, have globular non-collagenous domain. Okay, you should remember this name, globular non-collagenous domain. And it is of importance that sometimes antibodies in some autoimmune diseases, antibodies can be formed against this domain, and it leads to glomerulonephritis, which is anti-glomerular basement membrane glomerulonephritis. Okay, let's revise it. The collagen type 4 alpha peptide chains triple helix is forming the GMB, and it consists of a globular non-collagenous domain, and sometimes antibodies can be formed against this domain, so it results in the anti-glomerular basement membrane glomerulonephritis. Okay, and sometimes the similar domains are also found in the alveolar basement membrane, because the same type of collagen is forming the both of the basement membranes. So sometimes antibodies can attack both the lungs alveolar basement membrane and the kidney the glomerular basement membrane, resulting in a syndrome which is known as the good pastor syndrome. I don't know why they call it good, but indeed it's bad. But anyways, maybe after some scientists they name it good pastor syndrome. It affects the lungs and it affects the kidneys at the same time, and it results in hematuria. Hematuria means that blood in urine, and hemoptysis means that blood with cough, and when we cough there is the blood. Okay, it's hematuria and hemoptysis are the consequences of the good pastor syndrome. Okay, also one thing worth mentioning here is that this domain, which is the globular non-collagenous domain, is rich in polyanionic molecules. Why we need anionic molecules? Because the glomerular basement membrane has a negative charge. And why negative charge is important? Negative charge is important because we need to pull the negatively charged proteins in our blood. For example, albumins, the globulins, the fibrinogen. We not need them to be filtered in the filtrate, so they are negative, and the glomerular basement membrane should also have the negative charge. Like laminins and fibronectin, they impart electronegative charge to the basement membrane. Okay, the GMB, the glomerular basement membrane, should have the negative charge in order to repel the plasma proteins. Okay, this is also a diagram in which you can appreciate that this is the lumen of the capillary. These are the endothelial cells of the glomerular capillary. Okay, And in between the endothelial cells of the glomerular capillary, there are fenestrations about 70 to 100 nanometers. They have space, and anything in between 70 to 100 nanometer can easily be filtered. After that, this is the glomerular basement membrane. It has fibronectin, laminin, and other polyanionic molecules that impart negative charge to this membrane in order to repel the plasma proteins and other many essential proteins which are present in our blood. After that, these are the podocytes. The podocytes, visceral layer of podocytes, and after that, this is the parietal epithelial cells, and after that, it forms the proximal convoluted tubule. Okay. This is the Bowman's space. There is no fun in writing that. Bowman's space. Okay. This is the proximal convoluted tubule, which is formed by the parietal epithelial cells of the Bowman's capsule. And then is the glomerular filtration system. How this system functions, we'll discuss in detail in the next lectures, but here's some introductory concepts. Epithelial and fenestrated endothelial cells also have negatively charged molecules which are known as sialoglycoproteins. Epithelial cells of the Bowman's space and endothelial cells of the capillaries have negatively charged molecules, because I have already told you that we not need to filter the plasma proteins because they contain the negative charge. Okay, another filtration slit is coming, which is the present between the podocytes, the food processes of the podocytes. The slits are present interdigitating food processes of the visceral cells, which are the podocytes. In between them, filtration slits are present. These are not the slits between the endothelial cells, the first part. Second, the filtrate has to pass through the GBM. After that, these are the filtration slits. This is the third barrier which need to be crossed by the substances in order to become the part of the filtrate. Okay, these filtration slits are very, very narrow, about 20 to 30 or 20 to 40 nanometers. And these filtration slits have a protein known as the nephrin. Okay, nephrin, and antibodies may develop against this protein. Wherever there are proteins or any domain, and in autoimmune conditions, antibodies which are produced by our own cells can be developed against these proteins. So we develop autoimmune diseases like glomerulonephritis. So, in conclusion, the filtration in order to be formed has passed through three structures: the endothelial cells of the capillaries, after that the basement membrane, these are about 70 to 100 nanometers, the basement membrane. Filtration slits present between the food processes of podocytes are 20 to 30 or 40 nanometers in width. Okay, the factors in filtration are very important, and there are only two: the size and the charge of the molecule. When we increase the charge or when we increase the size, it is inversely proportional to the filtration. Increasing the charge or size decreases the ability to pass through the glomerular filtration barrier. If the charge is negative, and the glomerular basement membrane also have a negative charge, so it repel each other. Increasing the charge negatively, it decreases the filtration. But positive charge molecules, the cations, can pass because negatively charged basement membrane attract the positively charged molecules. For example, dextran. Dextran are the molecules which have variety of sizes and charge, so they pass accordingly. If the charge is suitable, is positive, can pass. If the size is suitable, so they can also pass. The blood elements, blood cells, for example, RBCs have 7 to 8 micrometers. It is a very enormous size as compared to the filtration barrier. WBCs are 12 to 14 micrometer. They are micrometer. Micrometer. They are not nanometer. Micro. And platelets are 1 to 2 micrometers. So these are not filtered due to the large size of these cells. The last but not the least concept is about the albumin. Albumins are plasma proteins and they have negatively charged. I cannot stress it more that albumins have negatively charged, but other plasma proteins, for example, the fibrinogen and the globulins, they have a large size, but the albumin is the smallest one. So why it cannot be filtered? It has the exact size, so it can be filtered through the filtration barrier, but it does not filter because it possesses a negative charge. Okay, it is not filtered in the healthy individuals due to repulsion forces. But in diseases like, for example, minimal change glomerulopathy, and in this condition, it is a minimal change disease. Minimal change, that the changes are not significantly more, but the visceral epithelial cells, which are the porocytes, they swell up. They swell up and they lose the food processes. Okay, and when there are no food processes, so the albumin can pass through uh, the filtration barrier and appears in the urine, and we found the condition known as albuminuria. Okay, after that, also there is other cause of the minimal change glomerulopathy, that some of the cytokines are produced in the body that neutralize the negative charge of the filtration system. As we have discussed in this diagram, that endothelial cells, the filtration slits, and the glomerular basement membrane itself contains molecules like sialoglycoproteins, like laminin, fibronectin, etc. They impart negatively charged to these filtration systems. And in the case of minimal change glomerulopathy, some cytokines in the body neutralize this negative charge, or it can be neutral, so the albumin can pass through this and appear in the urine. Mesangial cells. Mesangial cells have special features. For example, they can contract, they have receptors for the angiotensin 2, and according to the renin angiotensin aldosterone system we'll discuss in detail, it causes the vasoconstriction and it can result in hypertension. Mesangial cells can phagocytose the foreign, for example, anything, the bacterias or antigens. They can proliferate, they can divide, they can deposit the collagen and mesenchyme, the connective tissue, and they can produce biological products that can induce inflammatory response. So these are the special properties of the mesangial cells. I hope you got the concept of the filtration system and about the functioning of it, the charges and the size barrier of the filtration system, and which are the constituents of the glomerular basement membrane. So that's for today, and we'll discuss the other features of the glomerular filtration system in the next lectures. Okay, till then, goodbye.